what we can say empirically from science is that evolving the human condition is not easy, it's rare. I mean, this is a puzzle. What you're talking about is the human condition, right? So if we broaden human for meaning our species, but just think about, like, I was just talking with a theologian earlier this week about whether or not Klingons are human too, right? So Klingons, they're from Star Trek, right? They have the human condition, right?
Yeah.
They're not Homo sapiens. They're not Earthlings, but they share the human condition. They have a mind you can talk to. You get what I'm saying, right?
Yeah. Or the problem if we create a conscious AI, which I don't think we will. But
yeah, exactly. So here's the question: why is it that that the world we find doesn't have Homo sapiens alongside, Klingons, something with a different, intellect that arose that's just like us? That's a real interesting puzzle. I mean, similar to that, which you could ask is, the Jungle Book, it has like talking animals in it. Actually, a lot of our literature has like talking animals in it, right?
Or,
in Chronicles of Narnia,
all
these animals actually have the human condition, and in that sense, they are human. I mean, they're not Homo sapiens. I mean, but they're human, right? So it is a valid question. Why is that world that we can imagine very different than the world that we encounter, where animals are just not like that? I mean, there is no peer to humans in terms of that.
So what is your theory about how God intervened to make the transition? Let me say it like this. So we have a body that is, for some reason or another, capable of connecting to what we call our soul, so that we have a human condition.
What I would say is that what we can say empirically from science is that evolving the human condition is not easy, it's rare. So one way to think about it is that there were several lineages in the past that were related to, that had an opportunity to evolve the human condition, but did not. So we know, even starting from the starting point of, what our common ancestor was of the great apes, the vast majority of times, you're not going to end up with the human condition. A great example of this is actually, a great parallel to this is like Lenski's experiments when he looks at evolving citrate. So he has 12 lineages of E. coli, right, that are evolving. And only one of them evolves the ability to metabolize citrate. And when he's asked, well, what's the probability of this? He says, I don't really know. But provisionally, I guess I could say it happened 1 out of 12 times. So it's about 1/12. But of course, we don't really know because that's not enough data. It could be 1 out of 1,000,000 times and he just got lucky, right? And so it's the same sort of thing here. We only have a very, very small number of trials. We can't go back and do other trials. We know that one lineage found its way to the human condition. It could be a 1 out of 1,000,000 chance that happens, or it could be a 1 out of 6 chance, but it's not, we don't see it anywhere else. So there's good reason to think that it's not easy to do.
Let's expand our IPAN a little bit. So even if we say that because of the phenotypic similarity between humans and great apes, then all the marvelous designs in the human body can be, we can find similar things in our closest complex being, I don't know, the chimpanzee or whatever. But let's broaden it up when we are looking at real innovations, etc. And really the math does not. So we will look at molecular clocks which are based on neutral mutations. The question is always, how do you get the novelty and how do you get all the functions, etc.? In your opinion,
there's a lot of work on this. So so people obviously care about human evolution, and like 10 years, or actually it was 15 years ago, I read the human chimpanzee genome paper. Since then, we've learned a lot about human evolution genetics, particularly of the human brain, that we didn't know since then. Okay. So while it is that you can kind of lay the chimpanzee genome next to the human genome and you can see differences, you don't know how many of those differences or which ones of them are actually really important. That's actually why it's difficult, because of neutral theory. The vast majority you don't think are important. So how do you find the needle in the haystack there? So that's like a fundamental challenge, right?
But by definition, they are neutral, right? It's the neutral system.
Yeah, but then there's some that actually are not neutral. They're functional and they were positively selected, right? And so, but we've actually gotten a lot of more insight into that. So one example is the Duff family of proteins. And another, and I'm pretty sure this is one of the examples. I don't want to misstate it though. It's the RGAP, the RGAP 11 protein, I think it is. Once again, we've done this. I can send you links on this. It turns out that there is a single point mutation in a protein that's important in neural development. Okay. That's different between us by just a single amino acid change. I'm sorry, single nucleotide change. I'm sorry. Okay. All right. That alters a splice site and then adds on a bunch of DNA that's untranslated in chimpanzees, but now it's translated in humans. Okay. So all the great apes have this piece of DNA that's just not used. All right. But in humans, there's a one nucleotide change that causes that particular DNA to get turned on and actually added to the end of a protein. All right.
All right.
And so then you can ask, well, what does that do? Well, it turns out that, that's actually a question that biology can start to answer. So they've taken it and put it into other animals to see what happens if you change that one protein. Turns out that same protein is also in mice. And so you can take mice and then put in that mutation to add on that extra piece of DNA, the extra piece of protein, right? And see what happens. And what they're able to show is that actually does something surprising. It actually, that one change, that one mutation increases the amount of neuroprogenitors produced at a very particular point in development. Mm-hmm. And with having more progenitors there, then it ends up that the gray matter and the cortex ends up being bigger, and it goes from actually being a smooth brain to actually having sulcuses like ours.
Okay, so the folds would
with a single point mutation that causes not one, which takes previously unused, DNA sequences and accepts it or reuses it for the sense of a protein and does it. That's surprising. Okay.